All right, so last time we met, we had transitioned to talking about the organelles inside of the cell, to then talking about the plasma membrane, which functions for many things to give cells, or it functions in many ways, providing cells with their unique properties. Um, One of those functions is to move stuff in and out of the cell, which is absolutely essential to the life of the cell and the ability to survive. So really the majority of the rest of this lecture is going to be talking about cellular transport, bringing stuff in and out of the cells, which we classify as either active or passive. Passive processes involve substances moving down their concentration or electrical gradient. On the next page, we'll talk about what a gradient is here, so we'll get to that. This uses only their kinetic energy. When we use the phrase down their gradient or down a gradient, what we mean is going from high to low. Thinking of down a gradient like rolling a ball down a hill, which all it takes to get a ball down a hill is a little nudge, and then the ball's going to roll, or even something as big as a boulder, it's going to roll down the hill thanks to the energy of kinetic energy provided through gravity. Okay. This is how passive works. Active is the opposite. We're moving from low to high against a gradient. So when you say against the gradient, whether it's concentration or electrical, that's what active is, against the gradient, low to high. Think of this like rolling a ball up a hill, which requires your energy to do so, energy in the form of ATP, which we've talked all about already and we'll continue to talk about. Now there is another form of active transport that we'll talk about called vesicular transport that uses these bubble vesicles formed by the plasma membrane to either bring stuff into the cell or out of the cell. And we'll get to that towards the end of the lecture. First, let's talk about those gradients, though. There is 3 gradients listed on this page: concentration, electrical, and electrochemical. I'll explain all 3 of them individually. Let me let you guys write this stuff out, and then I'll explain. All right, so first for concentration, reading through the selective permeability. Remember, selective permeability refers to the plasma membrane only allowing certain things in and out. So the selective permeability of the plasma membrane allows a living cell to maintain different concentrations of certain substances on either side of the plasma membrane. For things that have no net charge, so things that are nonpolar, things like oxygen and carbon dioxide, they're going to move down their concentration gradients, which is what determines their movement. When I say down, I mean going from, once again, high to low. So let's look at our figure here on the left side of the page. We've got some molecules. Let's just call these oxygen. And let's pretend that in this region of our figure, letter A, we're looking at the extracellular space, so outside of the cell. Here's our plasma membrane that separates the outside of the cell from letter B, which is the inside of the cell. Now, you guys can go through and count out, but it's pretty obvious that there's a difference in number here and here. There's more in letter A than there is in letter B, right? If we are going down the concentration gradients, which direction are we going to go? When I say from high to low, are we going to go from A to B or B to A? A to B. That would be an example of passive movement, which does not require energy. We're always going to try to get the numbers equal, what we would call equilibrium. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 4. There's 14. We want to have 7 here and 7 here. And then there will be no net movement because it would be equal. What happens if we move against the gradient? We're going to go B to A, which requires energy. And we'll get to that today. Okay. Now, our plasma membrane also creates a difference in the distribution of charges. Remember we talked about positive ions and negative ions? Well, they're going to be located in different numbers inside and outside the cell. And for that reason, the cell will have a certain charge. Typically, the inner surface is more negatively charged. The outer is more positively charged. We can see that right here. There's not a letter A or B here, but this would be inside the cell, which if we stuck an electrode in there to measure it, we'd see that it would register as negative because there's more of these negative ions inside, more positive outside. Okay? This difference in electrical charge between the two regions, inside and outside the cell, constitutes its electrical gradient, which we term its membrane potential. Okay? And for most cells, their resting membrane potential is around negative 60. Some means they have a negative 60 charge. We'll talk about membrane potentials more in the next slide. Okay. Now, when ions move, since they are charged or polar, they're going to be moving based on its electrochemical gradient, which is based on the electrical gradient and concentration gradient. Okay. So let's look at our figure. With all these negative ions inside the cell, very few outside, so let's just put them as chloride. Let's just call them chloride ions. There's a high concentration of electrical, or high concentration of negative chloride here, very few here. So which direction are we going to want to go if we're going down the gradient? High to low, A to B, if it's letter A and letter B. Okay. What's going to happen to our charge when that occurs? We're negative, but we lose negative ions. So we're going to become more negative or less negative? Less. Exactly. This is absolutely essential for action potentials, for nerve signals, and this is what we're going to talk about next week, which is nice because we can transition from talking about it here to learning what actually is occurring. And you guys are going to love it. It's exciting stuff. We've seen this term, membrane potential, just recently. But this results from an excess of negative charge on one side of the cell, the plasma membrane, and an excess of positive charge on the other. At rest, cells are normally going to have a resting membrane potential. We abbreviate V sub M. V is short for voltage, M is short for membrane. Okay. We call it resting membrane potential because it's its charge at rest. Okay. And we can measure that, like I said, if you have an electrode inside and an electrode outside the cell, you'll find that inside the cell it's going to be more negative. Typically, cells are going to be negative 60 for their resting membrane potential. Negative 60 millivolts is what the MV stands for. Okay, not that that's important. Okay. Now, some cells will have a greater negative, like some might be negative 75, okay? Some might be less. But for the average cell, it's about negative 60. Okay. Electrical excitability is the property of a cell to rapidly change this charge. So it changes its membrane potential as a result of a stimuli trigger. This is what happens when we create an action potential. Okay. Ion channels will open up. Ions can flow in or out. The three major ions that we will talk about in this class are shown down here at the bottom: potassium, sodium, chloride. They even tell you where they're highly and less concentrated. Potassium is highly concentrated inside of the cell, low outside. We can see that over here as well. So which direction does potassium want to go? From inside out, right? Now, if that happens, what happens to our negative 60? Is it going to become more negative or less negative? Sarah, say it louder. You're just guessing, aren't you? What charge does potassium have? K plus. So if we are losing positive charges, we're going to become more negative. Because the positive charges that we do have start to counteract the negative. Well, we're losing those positives, so we're going to go this way. Sodium does the opposite. Sodium wants to come in. Sodium has a positive charge. So this number is going to become less negative. It might even, if we have enough sodium rushing in, go past 0 and become positive. These two are the main ones that we talk about when we talk about action potentials. Now chloride is a lot like sodium because it's wanting to come in. But what charge does chloride have? Negative. So if chloride comes in, it brings negative charges, we're going to become more negative. Okay. We'll talk a little bit about chloride. Chloride actually acts to stop nerve signaling. It inhibits the nerve signal. We'll talk about like reflexes, how you kick your leg out when you tap on the knee. We'll talk about how chloride is involved in that and how inhibiting action potentials is important. So I told you it was fun, right? All right, what we're going to do is go through and learn about the different ways that stuff come in and out of the cell. The first type of movement is called diffusion, and there are several forms of it. The first type that we'll talk about is simple diffusion. This is the easiest, it's the simplest. Okay? This is how small nonpolar molecules like O2 and CO2 get into or out of the cell. Literally, it's like a ghost floating through a wall. Instead of a ghost, it's an oxygen molecule or carbon dioxide, and it's direct and unaided. So nothing has to help it. It's like floating through a wall, which we can't do, can we? Okay, if you could, you wouldn't be in this classroom. You'd be doing something cool. We're going directly through the lipid bilayer based on the gradient, whether it's concentration. Well, it is concentration since it's nonpolar. Okay. So like from high to low, what I was just talking about. Always towards equilibrium. And simple means that we don't need transport proteins. We don't need a door or doorway. Would even be better than that. Things that can do this include oxygen, carbon dioxide, but other things like nitrogen gas, fatty acids, steroids, fat-soluble vitamins. Remember the other day how I said that fats can move through fats? Okay. Well, fatty acids are obviously fats. They move through the phospholipid bilayer pretty easily. So do steroids. What are steroids? Fats. And then vitamins A, D, and K are fat-soluble. They're just like fats. Fat-soluble means that they can pass directly through that fatty phospholipid bilayer. For most things, though, just like us, we can't get in and out of rooms directly through the wall. We need something that allows us in or out, like a doorway or, in this case, a transport protein. The concept is still the same. We're going down the gradient from high to low. This could be concentration, electrical, or both. We call this facilitated diffusion. You guys know what the word facilitate means? It means to help. So this is a form of diffusion that requires help. The other name for this is passive transport because it requires no energy. Energy in the form of ATP. No ATP is needed for this to occur. Okay. Now, simple diffusion is also passive, but we don't necessarily call it passive transport. We just call it simple diffusion. There's two different forms of facilitated diffusion. All it means is the only difference is who's doing the moving, whether it's a channel or a carrier. And we talked about channels and carriers just the other day. Remember, a channel is like a doorway that as you move through it does not change its shape. This is how most ions get in and out of the cell. Notice how I wrote channel in there with bold, and then over here channel is in italics. That's the difference between carrier-mediated, which moves it using a carrier. Okay. Both go down their gradients. It's just how they get there that's different. Okay. Here on the next page we can show you the image to show it off. Channel-mediated, the protein never changes shape. Carrier-mediated, the protein does change shape. Okay. Like I said, ions move through channels, so most channels are ion channels, allowing for the passage of small inorganic ions. Too hydrophilic, means that they like water. Remember that the tails of the phospholipids don't like water. They're hydrophobic. So that's how ions can come in. Sodium, potassium, chloride that we just talked about use ion channels. There are specific ion channels for each one of those. Some of them have a gate. Just like that doorway there that has a door covering it, they only open up in response to a specific stimulus. We're talking about ligand-gated channels. They open up when a chemical binds to it. Voltage-gated channels open up when a specific voltage inside the cell is met. This is a very fast process. Example there, more than 1 million potassium ions can flow through a potassium channel in 1 second. It's an incredible amount. Carrier-mediated is where the solute binds to a specific carrier on one side of the membrane, released on the other side after changes occur to the carrier protein. Rate at which this form of facilitated diffusion occurs is determined by the steepness of the concentration gradient across the membrane. So it's dependent on how much difference there is. Okay. Easy example I was just talking about this morning. If we use numbers to help, this should really ring true. Okay. So let's pretend on one side of our membrane, we've got 10 molecules of whatever. Okay, glucose, we'll just use glucose. Okay, on the other side, we've got 50. Difference between 50 and 10 is 40, right? Okay, what if instead of having 50, we've got 100 and still having 10? There's a bigger difference there, right? Now the difference between the two sides is 90. The bigger the difference between the two sides, the faster things diffuse. Okay. Think of it just like the steeper the hill, the faster the ball rolls. At 50, the hill is not very steep, but at 100, the hill is really steep. The ball rolls a lot faster. Does that make sense? Okay. So that's how speed of diffusion for carrier-mediated is regulated. Okay. So here are the 3 forms of diffusion we've talked about. Simple is real simple. No proteins needed. Channel, we're going from high to low through a channel. High to low for a carrier. This is glucose, by the way, moving through a carrier where the protein changes shape. Always, once again, going down their gradient, high to low. There is a 4th form of diffusion, specifically for water, called osmosis. This one's pretty cool, very important. As we know, there's a lot of water inside of our body. So this one is constantly occurring as we flow water around. So osmosis is just the fancy way to describe water diffusion. It's the same concept. We're going to go from high water to low water. Okay. And when water moves across the membrane, it does so through what we call aquaporins. Aquaporins are just simply water channels. Okay. In order for osmosis to occur, there must be a concentration difference across the membrane, which results from what's dissolved in the water. Remember how we talked about water likes to dissolve things? Okay. Those things that are dissolved in water are solutes. Okay. And water, well, let me just show you on the next page. We have an example here of how osmosis works. The same thing is basically at the bottom. That's kind of like this one. It's a little bit better shown. We've got solution 1 and solution 2, with solution 1 containing 5% albumin, which is a protein, and 10% albumin. Now, what we're talking about is not solution 1 here, solution 2 here. This is solution 1, this is solution 2. Those are separated by something that's a lot like our plasma membrane. Okay, this is actually a test you can do in a lab. Okay, now you guys would all agree with me that the volume between the two sides is the same, right? The amount of fluid is the same on both sides, right? Okay, over here it is. Now after time, it's not. But the amount of water is different between the two sides. Let's just pretend that instead of saying this is 5% albumin, we could say this is 95% water, and this is 90% water. So there's more other stuff over here, right? So this is high water concentration. This is low water concentration. Look at the direction that the arrow is pointing. There's more going this way than this way. So over time, what will happen is the volume of solution 1 will drop. The volume of solution 2 will go up, which helps to balance out the percentages for the albumin dissolved in it to achieve equilibrium. So you can describe osmosis as going from high water to low water concentration, which is down its concentration gradient. You could also describe it as going from low solute to high solute. So it's kind of backwards how you would think of it that way. Okay. Low solute would be where there's less other stuff. Higher solute would be where there's more other stuff. And so we go from low to high. Now as that occurs, the volume will go up. Okay. But are cells going to have an infinite capacity to fill with water? The answer is no, because just like a balloon, that if you fill with water, there's going to be a resistance to filling up, because just like a balloon, there is a boundary on the outside, which is the plasma membrane. The more water that fills up, the greater the amount of pressure that builds inside. This pressure that we're talking about is called osmotic pressure. The pressure that develops as a result of osmosis. Okay. And so when we go across the membrane, the pressure will go up. Osmotic pressure is important for a few different things. Let's talk about tonicity and how that plays a role with osmotic pressure. And volume as well. Okay, so explaining tonicity. As water moves by osmosis into or out of cells, their volume will increase or decrease. Okay, it's either going to fill up or it's going to spit water out and decrease. A solution's tonicity is a measure of the solution's ability to change the volume of cells by altering their water content. We can describe solutions as iso, hyper, and hypotonic. And we've got an image for each of those at the bottom. What we're talking about is the solution of the fluid inside of our cup. And these red circles are red blood cells. Okay. If we put a red blood cell in a isotonic solution, the red blood cell is not going to change shape because isotonic solutions have the same potential osmotic pressure because they have the same concentration of impermeable molecules. What I mean by that is basically what's dissolved in here and this solution as a whole is essentially the same thing as the solution inside of the cells. So water moves in and out at the same rate. So nothing changes to the cell as far as its shape goes. Hypertonic, which hyper means high, hypertonic solutions have a high level of solutes, which causes the cells to lose their water. It drains the water out to try to balance it out. We call this process where cells shrink crenation. Now, it's common knowledge that if you get stuck in a lifeboat in the middle of the ocean, what is the last thing you'd want to do? Besides jump in the ocean and swim with the sharks. You don't want to drink the salt water, do you? What does salt water do to you? Dehydrate you. This is why. That salt water is hypertonic. It will get into your bloodstream, and then your body is going to lose all its water from the cells. All the water from your cells will go into your blood plasma, and all your cells will become dehydrated, and then they start shutting down. So you die quicker if you drink salt water than if you just don't drink anything. Hypotonic solutions are the opposite. It's like filling up a balloon with too much water and it causes it to pop. Okay, that popping is called lysis, or hemolysis in this case for red blood cells. Okay. They have basically nothing in them, so that pushes the water in. So hypotonic solutions like just basically pure water. Nothing else in it. Why this is important? Well, some of you guys might want to be a nurse someday. A job of a nurse a lot of times is to give a patient an IV. IV is a solution of fluid that hopefully is not going to cause their cells to burst or their cells to shrink. So this might be something that you will have to think about later in life. Now, there are several factors that can affect diffusion rate. We've already talked about one of them, and that was the first one listed here on the bullet, describing the steepness of the concentration gradient. The steeper the difference, or the greater the difference between the two sides of the membrane, the faster the rate of diffusion. Okay, like having 100 on one side and 10 on the other side versus 50 and 10. Okay. Temperature actually plays a role. This one's something that can be easily demonstrated with 2 cups of water and some food coloring. You may have had a teacher do this before. Okay. Hot water cup and a cold water cup. If you drop a drop of food coloring in each, the food coloring will eventually spread out, cause the fluid to become blue or red or whatever. It's going to do it a lot faster in the warmer water because the warmer water allows the molecules to move around faster. The mass of the diffusing substance plays a role. This one is easy to describe. Basically, the bigger the substance, the slower it moves, which makes sense. Okay, the bigger we are, the slower we generally move, right? Okay. Surface area, the more surface area available for diffusion, the greater the rate of diffusion. Okay, how we talked about the other day, microvilli illustrates this. Remember, microvilli had little finger-like extensions. It means that there's more places for stuff to come in and out, so faster diffusion rate. Okay, places like our lungs have a huge amount of surface area because we need to diffuse oxygen, CO2 in and out. And then diffusion distance. Okay, the farther the place or the substance has to diffuse, the slower it is. Okay, the farther you have to walk, the slower you're going to get there, right? Take 10 steps or take 2 steps. Which one's going to be faster? 2, right? Fortunately, where we have to do diffusion really fast, like in our lungs, this is very minuscule. Like to get oxygen from our lungs into our blood, it's like 0.004 millimeters. It's like nothing, like paper thin. Okay. Guess what happens if you smoke though, and you get lung diseases? It's going to start thickening up. So you get scar tissue and stuff building up in there. So don't smoke. If you do, stop. All right, we're going to move on now to active transport. Active is where polar or charged solutes enter or exit the cell against their gradient. They're moving uphill, which we said requires what? ATP, which is our body's energy. Okay. The other name a lot of times for active transport is just pumping, okay, or other way to describe it. And pumping is really mostly specific to the first type here listed, primary active transport, which we're going to talk about first, then we'll talk about secondary. Primary active transport requires energy from the hydrolysis or breakdown of ATP. The best example of this is down at the bottom with the protein known as sodium potassium ATPase pump. Notice how I said pump because that's the other name for carrier proteins involved in this process. And this process moves 3 sodiums against their gradient from low to high, and potassium against its gradient, 2 of them from low to high against their gradient. It's a 4-step process shown down at the bottom where we take 3 sodiums and push them out of the cell, which is against what they would normally want to do because there's more sodium outside the cell than inside. So instead of going from high to low, we're going from low to high. Okay. That's capable of happening because here we see in step 2 ATP is used. After the sodiums are expelled, potassium combines to that carrier. The carrier changes shape again and pumps those 2 potassiums. And then we can start the process over again. Potassium, just like sodium, moves against its gradient from low to high. Now, let me show you some numbers, or use the numbers to kind of illustrate why this is going to help the next thing. So let's use sodium, and let's just say 10 and 5. Okay. If there are 10 sodiums here, there are 5 here, and we pump 3 out, now our numbers become 13 and 2, right? 10 plus 3 is 13. 5 minus 3 is 2. Okay. What happened to the steepness of my gradient? Has it gotten steeper or less steep? It's gotten steeper, which means that there's an even bigger drive to push sodium in, right? That's where secondary active transport comes in. There's an even stronger push to get sodium in, which is how we power secondary active transport. As it states here, secondary active transport indirectly uses energy from the hydrolysis of ATP. Look down at the figure, we don't show ATP at all. The ATP that drives this happens in primary. For secondary to happen, primary has to happen first. In secondary active transport, a carrier protein will simultaneously bind sodium and another substance, change its shape. That allows both substances to cross the membrane at the same time. Now, that means that they can either come in together or one can come in while the other goes out. Okay. Symporters are like giving somebody a piggyback ride. Okay. If the only way you could leave this room was a piggyback ride given by me, That's what symporters do. Sodium, which now really wants to get into the cell, even more so than before primary, can come in. Glucose can piggyback ride with it. So can amino acids. When we eat carbs and we absorb glucose, that's how glucose actually enters our body from our intestines. We'll talk about that later in the semester. Okay. Now, antiporters move them in the opposite direction. So it'd be like me going out the door, leaving the room, somebody at the same time comes into the room. And here we see sodium coming in, potassium going out. Sodium coming in, hydrogen ion going out. Now, sodium is the linchpin in this case. Sodium doesn't always have to be the linchpin. Okay? Hydrogen ions can play a role in secondary active transport. Okay? Because hydrogen ions can also be involved in primary active transport. We just use sodium, potassium, ATPase pumped because that's the main one that we'll see in this class. And we'll see it as soon as next week when we talk about action potentials because it's involved in that as well. The last form of active transport, you don't really see ATP directly, but it is technically classified as active transport. And this involves vesicles, which are these like bubbles that either bring something into the cell or move it out, or a combination of the two. Endo, you can think of as bringing in or inside. Okay. Endocytosis is where materials move into a cell in a vesicle formed by the plasma membrane. Okay. Here's a picture that shows endocytosis. The membrane folds in on itself and eventually wraps around the substances and brings them in, forming a bubble known as a vesicle. Okay. The opposite of endocytosis is exocytosis, where the membrane-bound bubble fuses with the plasma membrane, the substances are released. Okay. Now, if we do both, bring something into the cell on one side and then spit it out on the other, we call that transcytosis, which is a combo of the two. Okay. There are several forms of endo. The ones that we talk about the most, or the one that we talk about the most, is phagocytosis. That's what's shown over here on the right. This is how, like, some of our white blood cells can kill bacteria and viruses. It's basically cellular eating. I like to think of it like Pac-Man. Pac-Man that runs around and chews stuff up, right? Okay. They swallow in the substance, forming what's called a phagosome. Remember lysosomes we talked about last time are those digesting enzyme or digested enzyme-containing organelles. Those things fuse together. The enzymes digest the substance, whether it's bacteria, virus, whatever, and then that thing will probably get spit out by exocytosis. Okay. There's also penocytosis. Instead of cellular eating, we call that cellular drinking. Penocytosis is the same thing as bulk phase. Okay. Tiny droplets of extracellular fluid are taken up this way. We'll talk a little bit about that later in the semester, way towards the back end of the class. And then the other form of endocytosis is receptor-mediated, which means that the substances that are going to be brought in must bind to specific receptors before they'll be pulled in. Okay. Like I said, the opposite of endocytosis is exocytosis, and then a combination of the two is trans, which means across or through. Okay. On one side of the cell, they go into the cell as endocytosis, moves across, spit out on the other side by exocytosis. Okay. We'll talk a little bit about that later in the semester as well, but not very much. Table 30.1 from your textbook, just kind of a nice summary of those various things that we just talked about. Last thing I want to talk about is the cell cycle and cell division. If you've taken biology, even back in high school, you should have learned about. We're not going to really talk a whole lot about it, and we're not going to talk about both forms of cell division. We're only going to talk about mitosis. Later, when we get to the reproductive system, we'll talk about meiosis, which is how men form sperm, females form eggs. Okay. Cell division is something that most of our cells go through. Cell division is just the process in which cells reproduce themselves, going from 1 to 2, 2 to 4, on and on and on and on. Cell division takes place as part of our cell cycle, which is an orderly sequence of events by which a typical or somatic cell duplicates its contents and divides in two. Somatic just means it's non-reproductive, so it's not an egg or sperm. Okay. The two primary phases of the cell cycle are interphase and mitosis, or mitotic phase. Okay. Here we can see a description of the two phases. Most of the cell's life is spent in interphase. This is when the cell's normally functioning, doing the different things that we talked about those organelles doing. Okay. There's G1, S, and G2 phase. Of those three phases, S phase is the most important to the cell division stuff because in S phase, DNA is replicated or duplicated. We take a DNA molecule and we unzip it. Remember, DNA is double helix, so two strands. Each of those strands gets attached another strand. And so what we end up with are two hopefully identical copies of DNA. By the end of interphase, the duplicated DNA inside of the nucleus is ready to be split so that you end up with two identical cells, each containing the same number of chromosomes. How many chromosomes do our typical cells contain? You guys remember? 46. Okay. That is accomplished by mitosis, which is where the nucleus divides, which also most of the time involves cytokinesis happening at the same time towards the end, which is where the cytoplasm divides. I always tell students this is how I remembered it. The correct order is just think of PMAT. Prophase, metaphase, anaphase, telophase. Here on this page, I explain very briefly what happens in each of the phases. There will be at least one question on these. And I might ask you, which following phases involves the nuclear envelope breaking down, chromosomes appearing, yada, yada, yada. Okay. So I'm going to just kind of back and forth bounce between this page and the page where I have pictures to show them off. Okay. Prophase can actually be divided into early and late prophase. Okay. Early in prophase, we see a nuclear envelope still intact. By late prophase, it's basically disintegrated and the nucleus has disappeared. Okay. We see the chromosomes as what we call sister chromatids. Okay. One chromosome is just easily draw it. One chromosome would be that. But since they've duplicated, they kind of form this X shape. Okay. Now, when they separate, they'll separate like they were when they were drawn by themselves. They're identical. This strand here is identical to that strand. Okay. So we can see those sister chromatids formed, okay, identical to one another. Now, at this point, the chromosomes, those sister chromatids, are kind of moving toward the center of the cell. They're not completely perfectly down the middle yet. We do see those little spider leg looking like structures, those microtubules that we talked about the other day, starting to move towards those sister chromatids. Metaphase, I always tell students, remember metaphase, which starts with M and middle, because that's where you find those chromosomes, right down the middle of the cell on what we call the metaphase plate. On the previous page, I think, yeah, they were in the center of the cell or middle of the cell. Here, as well, we see those spindle fibers or microtubules completely grabbing the waist of the X chromosomes. Okay, those chromosomes, not X chromosomes, but the X-shaped chromosomes. Anaphase, they're pulled towards opposite sides. the cell begins to elongate. Okay, early, then late. Okay. We might even get to see the development of a cleavage furrow, which is really evident generally in telophase, which is now when we start to actually see the cells start to split. That's the beginning of cytokinesis, when the cytoplasm divides. Telophase, those tubule or microtubule spindles will kind of go back and disappear. The nuclear envelope will start to form, the chromosomes will unwind, and by the end, after cytokinesis has taken place, which generally happens and finishes off in telophase, we're left with two identical cells, with each having a nucleus with the unwound DNA in it. Okay. Now, the way that I put this picture or this slide together is kind of misleading. It kind of looks like a cycle, but notice that there's not an arrow from telophase straight into prophase. From prophase, we would go in, or from telophase, excuse me, we'd go right into interphase, and then around back to prophase. So that's why there's not an arrow going from here to here. For us to survive and develop, repair, we have to have cell division occurring. Okay. Unfortunately, cell division can get out of whack sometimes. And I'm sure you've all heard of and known people with cancer. Cancer occurs when cell division isn't occurring correctly. This is where we have uncontrollable or abnormal cell division, which means that all of a sudden we've got all these cells being formed, and those form a tissue we call a tumor, or a fancy way to describe tumor is a neoplasm. You can classify tumors two different ways. Cancerous are described as malignant. Okay. Now, not all tumors really have this capacity or capability, but many are capable of performing metastasis. I'm sure you've probably heard that word before. If you aren't familiar with what that term means, it means that the tumor can spread. And so the cells will break free from the original tumor and lodge themselves somewhere else, forming a new tumor somewhere else, which makes tumors that metastasize really dangerous. Breast cancer is one that does this really rapidly, okay, which makes breast cancer so dangerous, especially in females where you see breast cancer and more incidences. Benign tumors, I always tell students the phrase, benign, you're fine. Now, that doesn't mean if you have a benign tumor that it's all hunky-dory. Benign tumors can be dangerous because they can impede and be in the way. But these don't metastasize, and they're technically not cancerous. Cells of malignant tumors will duplicate rapidly, continuously as their cells invade surrounding tissues. They often trigger angiogenesis, a process that is where they grow their own blood vessels, and then that means they're just going to get bigger and worse. Okay, and that can compete with normal tissues for space and nutrients, often causing death of the normal tissue to occur. And that's what causes the body to shut down when tumors develop and get so big that they shut things down. So, unfortunately, cancer is very rampant. There's a lot of cool treatments out there, radiation, chemo, I'm sure you're all aware of those. Angiogenesis is something that some treatments try to cut out. If they cut off blood supply, the tumor will die off. So there are some medications that will try to stop angiogenesis. One of the coolest ones that I've seen is where doctors are now injecting like polio into the tumor, which we all know polio is bad. It's a virus. But when you stick it into the tumor, it actually kind of activates the immune system of that person to go attack that tumor. And so it's been, I watched 60 Minutes on this about a year ago. That was pretty cool. Hopefully, something that continues to develop and we find a cure for cancer. All right, you guys got any questions for me?